far cost action, coordinated by Professor Antonio Quadrato from the Autonomous University of Madrid. I would like to sincerely thank Antonio for his continued support and dedication to this initiative, and we are grateful to him for kindly leading today's session. I also want to extend my thanks to the organized committee of the high-level webinar series, in particular to Professor Luciano Saso from Sapienza University of Rome, who has spearheaded the entire series with a great commitment. We are also pleased to share that a new webinar will be launched next year on the antiox.it platform. We warmly invite all of you to suggest topics and speakers for these upcoming events. Your idea will help us shape the program and sure we continue to highlight the most relevant, exciting development in the oxidative stress and NRF2 research area. And now it's before introducing our highly estimated speaker, who will be joining us in today's meeting with their experience and knowledge in the field of NRF2 activation and non-communicable diseases, I want to remember that each of the speakers will give a talk of about 25-30 minutes. The webinar will last up 2 hours. The discussion will be opened after all talks. Please feel free to ask questions in the chat at any time. And when doing so, please don't forget to mention your name and affiliation and to whom the question is addressed. Of note, the webinar will additionally stream it online upon registration. I'm really looking forward to hear all your talks, and I think it will be a very rich, interesting session. It's my pleasure to introduce our first speaker, Professor Christina Morgenstern, a molecular biologist and bioinformatician based at Vienna Airway Lab, Medical University of Vienna. Her current research focuses on biomarker discovery and understanding the NRF2-mediated redox inflammation axis in chronic respiratory diseases, combining high-throughput and multimodal approaches. Within the cross-section, Madam Christina has been a driving force leading Working Group 3, which focuses on the identification and validation of NRF2-related biomarkers. Today, she will present a lecture entitled Monitoring NRF2 Signaling, a candidate biomarker panel, its a clinical application. Please join me in welcoming Professor Christina Morgenstern.
Thank you, Brigitte, for the nice introduction. And welcome, everybody. Hello from Vienna. It's very kind of you, Brigitte, that you announced me as a professor. I'm not a professor yet. I'm working hard to make it to that position, but it's still some time ahead. But nevertheless, there's something to anticipate for my future. It's a pleasure to be here today and present the work of Cost Action BAM-BAD-FAR Working Group 3 Translational Medicine. Over the past 4 years, our mission has truly been to bridge the gap between foundational NRF2 research and its clinical application. And a central challenge in this effort has been answering a deceptively simple question: how can we reliably measure NRF2 activity in a patient? And today, I will present you our group's consensus roadmap on how to tackle this question and to propose a biomarker panel designed to solve this problem. Our work that I'm going to present has also been comprehensively described and in 2 recent publications in redox biology. But before we are discussing monitoring NRF2, let's briefly recap NRF2 biology. Nuclear factor erythroid 2 like 2, or NRF2, is a transcription factor that is encoded by the NFE2L2 gene, and it plays a pivotal role in the maintenance of cellular redox and metabolic homeostasis, as well as in regulation of inflammation and cellular detoxification pathways. The transcriptional activity of NRF2 is regulated through its binding to KEAP1, which presents NRF2 for ubiquitination by Calin-3 and Ringbox1 protein, and subsequent degradation through the proteasome, as seen on the left side of this cartoon here. Thus, under basal homeostatic conditions, NRF2 levels are very low within the cytoplasm. But upon modifications of key cysteine residues of KEAP1 by, for example, electrophiles, reactive oxygen species, or through disrupting the protein-protein interaction, NRF2 gets liberated and newly synthesized NRF2 accumulates within the cytoplasm, is then able to translocate to the nucleus and activate target genes through binding to antioxidant response elements, ARE sequences, together with proteins of the SMAF family. In that way, NRF2 can orchestrate its stress response. Talking about oxidative stress, which is broadly defined as an imbalance between pro-oxidants and antioxidants, and can be associated with the disruption of redox signaling and macromolecular damage. Persistent oxidative stress plays an important role in the initiation and progression of many chronic diseases. And altered levels of NRF2 activity have been inherent in diseases such as metabolic diseases, respiratory tract diseases, central nervous diseases, and cancer. We call these diseases non-communicable diseases because their causes are multifactorial and not caused by infectious agents. Those diseases progress often gradually, with symptoms worsening over time and persisting over years or lifespan, requiring constant management. This most often reduces the quality of life of the patient and might further lead to tissue damage and other secondary health issues. Thus, it is of great importance that we can track disease progression, monitor therapeutic outcomes, to bring down the individual suffering and also the economic costs. As NRF2 is a key regulator of oxidative stress responses, and this progressive cellular stresses and inflammation lead to chronic diseases, understanding NRF2 in this context is of uttermost importance. And to identify disease-specific or generic targets as a readout for intervening and restoring NRF2 levels could halt disease progression. NRF2's role in human chronic diseases can be viewed as a dual phase. In prevention and early on in disease, it's a guardian coordinating cytoprotection. However, in established diseases like cancer, its persistent activation can be villainous, promoting disease resistance and survival of damaged cells. This duality makes context-specific monitoring not just useful, but essential for developing safe and effective NRF2 therapies. There are a couple of drugs that have been licensed, such as dimethyl fumarate-based drugs for relapsing multiple sclerosis, or omaveloxolone for the treatment of Friedreich's ataxia, that intervened with this persistent oxidative stress and inflammation condition and might or functions through NRF2. But we need more drugs, and drug discovery is slowed due to the lack of biomarkers, which can inform target engagement as well as the magnitude and duration of action. Move on. So NRF2 can combat redox stress through modulating endogenous antioxidant response pathways, such as the detoxification of reactive oxygen species through superoxide dismutase and catalase. It is also able to impact, for example, the detoxification of quinones through NQO1. But NRF2 is also able to modulate intermediary metabolisms, such as lipid metabolism, but also is implicated in proteostasis. Moreover, NRF2 can negatively regulate inflammation through the blockage of pro-inflammatory cytokines, such as interleukin-6 and interleukin-1 beta. So there have hundreds of targets of NRF2 being described in the literature, which makes a vast NRF2 regulon. While these targets reflect its central role in homeostasis, it creates a problem of choice for researchers and clinicians. Which of those targets provide a faithful and consistent readout of NRF2 activity? And there has been a lack of consensus in the field. And this gap, our working group has tried to close. And I'm now going to present our roadmap on how we defined a consensus biomarker panel to assess NRF2 activity. So, as mentioned before, NRF2 is known to regulate hundreds of genes in mammalian cells, but which ones are the most relevant ones? And we set out to define strict evidence-based criteria to define true activity markers of NRF2. We started off with a database search of PubMed and dedicated search terms and retrieved more than 200 papers that have measured NRF2 activity across different cellular contexts. We then extracted those data and came up with a candidate list of more than 1,000 target genes and proteins that have the capability of measuring NRF2 activity. We then defined and narrowed down those lists based on 3 criteria. First, a marker of NRF2 activity has to be a direct target of NRF2 through chromatin immunoprecipitation. Second, the candidate has to be significantly downregulated in response to inhibition of basal NRF2 activity. And third, the candidate has to be significantly upregulated in response to pharmacologic activation of NRF2. So to refine the panel of markers, we focused only on genes / proteins that satisfied all of these 3 criteria with evidence from at least 3 distinct publications for each of the criteria and spanning more than 10 different cell types across multiple species. And this rigorous triage ensured that we moved from a long list of biomarkers to a short list of markers capable of being an NRF2 fingerprint. And this is the result that yielded this 6-gene protein core signature: GCLC, GCLM, HMOX1, NQ1, SRXN1, and thioredoxin reductase 1. And these are not random choices. They are mechanistically linked to NRF2 functions. For example, glutathione synthesis, glutamate cysteine ligase modifier, and glutamate cysteine ligase catalytic subunits, GCLC and GCLM, they are participating in glutathione synthesis. HMOX1 or hemoxygenase 1 is an essential enzyme in heme catabolism. NQ1 or NADPH, quinone reductase, is essential in quinone detoxification. And we have sulfur-redoxin 1 and thioredoxin reductase 1 exerting antioxidant function. So as this string network shows, they form a functionally interconnected complex representing a cohesive fingerprint of NRF2 pathway activation. And so, sorry, it's a bit slow in moving the slides, my apologies, but yeah, no, it was too fast because this interactome is really, it's a heavy picture. Our panel of 6 biomarkers, it exists within a much larger network of NRF2 interactome, as you can see here on the left, or appreciate all the dots that are proteins more or less densely connected within the center and loosely connected outside. These are the more than 1,500 proteins that we retrieved from our search, and the core 6 proteins that emerge here as green dots with NRF2 as blue dot here at the center. And if we subject those proteins to pathway enrichment analysis using the Reactome database, we can be assured that those proteins, they are implicated in the NRF2 pathway, as seen here by the evidence, or in oxidation and other metabolic processes. So this confirms that our 6 marker panel sits at the very core of this NRF2 pathway. But this brings us to the question, is this fingerprint of 6 genes proteins actionable also in a clinical setting? Can we move from a tissue-specific analysis to a systemic readout that can inform the status of a patient? And our further investigations set out to answer whether this panel of 6 markers can be used to monitor the state of NRF2 signaling across different non-communicable diseases in clinical settings. And instead of locally monitoring these markers, which can be challenging because some tissues might not be accessible, such as the brain, it would be interesting to trace a systemic readout of NRF2 activity in the periphery that can be used to assess the state of NRF2. And data from the Human Protein Atlas provided a promising start, confirming that all 6 markers are robustly expressed in various circulating immune cells. So on the transcript levels of these markers within the 6 immune cell types revealed that, for example, HMOX1 is highly expressed in monocytes, whereas NQ1 is highly expressed in natural killer cells. Conversely, the protein expression here on the right side, HMOX1 again is highly expressed in PBMC, so peripheral blood mononuclear cells, but also thyroidexin reductase and NQ1 are expressed within PBMCs to some extent. So this was some key insight and confirmation that peripheral blood draws could potentially provide a window into systemic NRF2 activity. To validate this systematically, we conducted an extensive literature review using the PRISMA, so the Preferred Reporting Items for Systemic Reviews and Meta-Analysis guidelines and outline, and screened over 580 publications that we retrieved with our search terms against stringent inclusion criteria, and ended up with 36 studies that we included into further validation. So this allowed us to aggregate some real-world evidence on how our panel might behave across a variety of human studies of non-communicable diseases. Our search revealed that the 6 markers have been measured across a vast spectrum of non-communicable diseases, from Alzheimer's to cancer. We have binned those 20+ diseases into major groups, so using their parent disease term according to the medical subject heading, MeSH terms. For example, Alzheimer and Parkinson's disease were grouped into neurological disease to assess the universal relevance of our NRF2 markers across different disease groups. Concerning sample types, we learned that peripheral blood mononuclear cells, or PBM cells, were the sample type that was most commonly used to assess our 6 markers. And this was followed by other circulation-related samples, serum, plasma, but also specific blood immune cell types like monocytes and leukocytes. PBMCs are great. They are accessible, easily accessible. They are minimally invasive biofluid and can be a very potential source of biomarkers that can be readily accessed. and objectively monitored for surrogate endpoint of NRF2 activation. So this proves the feasibility of a minimally invasive monitoring strategy, and quantitative reverse transcriptase PCR has been the predominant analytical techniques that we uncovered in our studies. Okay. So normally studies, they are limited to a couple of samples, especially when we are dealing with patients. But in our meta-analysis, we could draw evidence from an incredible large amount of samples. And in this bar chart, you can see the analyzed cases and controls across the different disease groups, with cancer having the most number of subjects assessed. The cases, the controls, they are comparing, for example, pre and post treatment, healthy tissue adjacent to diseased tissue, and so on. While the different studies were of different types and also of qualities, we also employed kind of a scoring system for the different studies, with studies that performed their well-designed individual randomized placebo-controlled trial with the highest evidence. And we could uncover 2 studies, one within the metabolic diseases and one within the chronic kidney diseases, to be of that type. So this is the most important result. This is when we aggregated the data from all these studies. It shows on the x-axis our 6 markers, and on the y-axis the diseases. And in green, you can appreciate the upregulation of those markers, with the size of the bubble corresponding to the strength of the significance. So the larger the bubble, the more strongly this marker was expressed. So key findings are that, for example, we see our panel expressed varyingly throughout different disease categories. Most often, HMOX1 and NQ1 were measured across different disease categories and found to be upregulated. Especially within neurological diseases, we find HMOX1, NQ1, and thyroidexin-1 upregulated in Alzheimer's disease, I think, or is it Parkinson's disease? It's very small. I'm on my screen, my apologies. On the other hand, we also find the very same targets across similar disease categories, but in different diseases, and also in the same disease, downregulated. So there is some kind of discrepancy. We cannot, this approach doesn't assure that all the markers are consistently upregulated across certain diseases. No, we also find downregulation, or markers that have been measured, but as not significance was retrieved. So this kind of tells us that there is no single marker that is sufficient to explain or to guide a readout of NRF2 activity. We believe that the power lies within the mix and match, so taking several biomarkers. And this slide summarizes the overall evidence of strength. It shows that in several major disease categories, multiple markers from our panel are consistently dysregulated. And now we have aggregated all neurological, respiratory tract, and so on, all diseases together, and show which markers have been assessed in those diseases. And for example, within the neurological category, 4 markers have been consistently upregulated. So GCLC, HMOX1, NQ1, and sulfur-redoxin 1. And this is also true for cancer. We found 4 of our 6 markers consistently upregulated within that disease. So we suggest using this panel is a good choice, and that the combination of the different biomarkers can provide more confidence than relying on a single biomarker. Additionally, and maybe some also overlooked aspects that we uncovered within our search are that factors like diet, age, sex, lifestyle, the time of the day might influence NRF2 levels. There might also be individual differences between NRF2 levels, which makes it difficult to nail down to specific biomarkers that might be meaningful. So this means future clinical trials must carefully select patients, maybe whose NRF2 system is still responsive, but not already overwhelmed or burned out. We also present here a strategy for further validation of these 6 markers for true clinical translation. Those markers need further analytical validation, so we need to establish a test platform. Are we going to assess mRNA or protein levels? And then also performance characteristics. They might be different between the different markers, as well as between different individuals or disease settings. And also those markers then need to be validated in further external clinical cohorts. So there is a long road to go to make really clinically use of this biomarker panel, and to ultimately approve regulatory acceptance for a specific context of use, maybe for one disease context. So there's still an exciting journey ahead for our panel. Maybe some outlook or a note on how to take it further to the clinics. How can the static panel serve in dynamic monitoring? As I mentioned before, patient selection might be key. So using target population with a functional activatable NRF2 pathway. Then adopting dynamic longitudinal designs to be able to capture the kinetic response of the pathway. And then this is also something that we find is important. Don't limit to one marker, but make use of at least this 6-marker panel to validation as a fingerprint. And then to make sense of the biomarker signature, correlate those with clinical endpoints. And finally, context is also key. So there needs to be some standardization in sample type and also timing. And by following these paths, we might be able to transition from a static biomarker panel of these 6 to a dynamic tool for monitoring patient health and therapeutic efficacy. If you want to dive deeper into this topic or also see what we have found within the individual diseases, this has been comprehensively described in our tool reviews. The first one, Biomarkers of NRF2 signaling, describes our approach of narrowing down this panel of 6. And the recently published review on altered NRF2 signaling in systemic redox imbalance, insights from non-communicable diseases, highlights really the clinical utility of those 6 biomarkers across different disease settings. So please go and check it out. And with that, I'd like to say that this work is the result of a truly collaborative effort. I'd like to extend my sincere thanks to all the members of BAMBET-FAR Working Group 3, especially to the colleagues that are listed here. I'd also like to say thank you to Antonio for leading this action and for entrusting me with the role of a working group leader. I'd like to say thank you to Brigitte, my co-leader of this working group. And thank you for your attention, and I look forward to discussing this with you further. Thank you. Stop here and stop sharing.
Okay, thank you, Christina, for your interesting talk. Now let's move to the second speaker. And it's a great pleasure to introduce our next speaker, Anna-Liisa Levonen, Professor and Vice Dean of Research at the University of Eastern Finland, Faculty of Health Sciences. Professor Levonen is a leading expert in oxidative stress biology and the redox-regulated gene expression. Her research explored the gene regulatory mechanism activated by oxidative and electrophilic stress, with a particular focus on the KEAP1 NRF2 pathway. Over the years, she has made outstanding contribution to understanding how NRF2 activation influences the development of cardiovascular diseases and cancer, published influential studies that have shaped the field. Professor Anna-Liisa Levonen has been a key contributor to cost action BAMBET-FAR, leading efforts in Working Group 3, and as well as supporting multiple other projects within the action. Today, she will share her latest insight in a lecture entitled Biomarkers on NRF2 activation in cancers. Please join me in welcoming Professor Anna-Liisa Levonen.
Thank you, Brigitte, for this invitation and introduction. Can you see my slides now?
Yes.
Probably yes. And
yes.
Let's see whether they will change. Okay. Now I guess you see the next slide. So today I'm going to talk about the biomarkers of NRF2 activation in cancer. I will continue on what Christina just told about biomarkers, and have a little bit of a different perspective, or more, let's say, more focused perspective on biomarkers, specifically in cancer. And why we think that cancer is really important topic to study in NRF2 biology is that we all know that in normal cells, NRF2 is actually protective. It protects against oxidative DNA damage and carcinogenesis, whereas at in fully developed malignant tumors, in many cases, there's constitutively active NRF2, possibly through mutations in either KEAP1, NRF2, or COLIN3, which results in a protection against high endogenous levels of reactive oxygen species, and also induction of phase 2 and phase 3 enzymes that are involved in chemotherapy resistance. So basically, there are multiple different mechanisms by which NRF2 can be dysregulated in cancer. Here we can see that in genomic mutations, you have either mutations in NRF2L2 gene, which is encodes NRF2, or KEAP1, and how these are different is that in NRF2L2 you have gain of function mutations in either DLG or ETG domains of NRF2L2, and KEAP1, because it's a loss of function mutation, the mutation can actually reside throughout the gene. We have also multiple other mechanisms by which NRF2 can be dysregulated in cancers. I won't go into details, but just to mention a few, you can have a deletion of the entire region that in NFE2L2 that binds to KEAP1. You can have suppression of KEAP1 expression through promoter methylation, oncogenic upregulation of the transcription of NFE2L2, as well as copy number amplification, etc. So genomic mutations are not the only way to upregulate NRF2 in cancer, but in lung cancer, it's most certainly the most common way of upregulation. And as I mentioned, NRF2 is a really important regulator of drug metabolism, resulting in resistance to common chemotherapies. For instance, here in this figure, you see that cisplatin resistance is heavily dependent on glutathione biosynthesis, which is all NRF2 regulated, cysteine uptake through SLC7A11, and you have the conjugating GSTs and the pumps that then pump the conjugates outside the cells. So all of these are NRF2 dependent and thus important for resistance against chemotherapy. This is a closer magnification of the cancer-relevant mutations in NRF2 and KEAP1. And as you can see, there are specific hotspot mutations in NRF2, but also in KEAP1. You can see that although they are more spread out throughout the exome, you can have actually certain hotspot mutations such as R320 and R470. Our focus in my group is mostly in non-small cell lung cancer, and that is because it is actually #1 killer in Finland as well as globally, and one of the most common cancers both in Finland as well as in the world. And also KEAP1 and NRF2 mutations are particularly common in this cancer. So non-small cell lung cancer can be divided into lung adenocarcinoma and squamous cell carcinoma. And here you can see that there's a distinction between these two cancers, so that you can have, you usually have an enrichment of KEAP1 mutations in adenocarcinoma, whereas NRF2 mutations are more common in squamous cell carcinoma. NRF2 pathway mutations also co-occur with other pathway mutations. They are sort of not only usually with respect to NRF2 mutations, you have quite often pathway mutations also in PI3 kinase AKT pathway, and specifically not only lung squamous cell carcinomas, but also other squamous cell carcinomas have enrichment of NRF2 pathway mutations. Here, this is a global view of incidence and mortality of lung cancer worldwide. And you can appreciate that, as I mentioned, the prevalence on the left side is really the highest in lung, even more than breast cancer and prostate cancer, which are really common, especially in Finland. And in lung, it's overwhelmingly most lethal cancer. And the five-year survival rate in Finland is approximately 20%. It's gradually increasing a little bit, but still it's a #1 killer and very a poor prognosis cancer. If we look into the histology and oncogenic mutations in lung cancer, this depiction tells the story that actually most cancers, non-small cell lung cancers, are actually adenocarcinomas, and we know quite a lot of the mutational signature of non-small cell lung cancer is extremely variable, but there are certain mutations that are highly enriched and actually at the moment also targetable. For instance, in adenocarcinoma, you have EGFR mutations. There's targeted therapies for that. Also, KRAS has been recently been able to be targeted through KRAS inhibitors, and also some other less common mutations such as ALK mutations, ROS1 mutations. Those also have specific drugs, but there's a vast number of other or not identified mutations, and actually NRF2 KEAP1 mutations are within this slice here. And at the moment, it's safe to say that there are no specific inhibitors to that. We really need to work on that. But also NRF2 KEAP1 mutations and other NRF2 the ways to increase NRF2 activity results in resistance to other therapies, not only the classical chemotherapies, but also targeted therapies, and there's increasing evidence for that. So, so this was my background on NRF2 in cancer and also specifically in lung cancer. Next, I will go to biomarkers of NRF2 activation in a non-small cell lung cancer. Basically, the first example I have here is the DNA sequence analysis, mutation analysis, and it's actually quite commonly used for clinical diagnosis already for various reasons. For instance, with respect to EGFR therapies, you have to first assess that you have a mutation in EGFR, but it can also be used for other types of things. You can use it for diagnosis so that you can distinguish between the patients that have a better or worse prognosis, and also you can, with certain cases, can detect heritable risk of developing cancer. KEAP1 mutations from the tumor tissue has been used as a biomarker for a number of different studies, and this study actually is with the novel KRAS G12C inhibitor specific to that, and quite distinguishedly, you can see that if you have a mutation in KEAP1, especially with combination of SMARCA4 and CDKN2A mutations, they really drastically decrease the efficacy of this therapy. So you can use mutations in KEAP1 as a prognostic factor and in lieu of studying NRF2 activation. Also, checkpoint inhibitors are quite commonly used in non-small cell lung cancers and very many other cancers. And just a brief overview for those who are not very familiar with what they are doing. This is an example of PD-1, PD-L1 mediated checkpoint inhibition. So basically, cancers have aberrations that result in expression of neoantigens that bind to MHC1 molecules, and these are recognized by T cells as foreign. And so cells, the T cells then kill the cancer cell, and this is called immunosurveillance. But cancer learns to escape this immunodestruction, and these couples that are now in use in clinics are PD-1, PD-L1, and also another one, CTLA-4 mediated checkpoint inhibition, so that the T cell no longer will kill the cancer cell. And this won a Nobel Prize a few years back. So it has been shown, this is one of the studies, but there are many others showing that actually KEAP1 mutations, especially in combination with STK11 mutations, correlate with diminished efficacy of PD-L1 inhibition. So mutations in KEAP1 can also, you can use that to detect poor prognosis for these therapies. This fairly recent study showed that if you combine PD-L1 inhibition with CTLA-4 blockage, then you actually abrogate this inhibition. But these immune checkpoint inhibitors are really quite, they have a lot of side effects, and having two different drugs is actually very, very hard for the patient. So clearly, although you can use these mutations as biomarkers and then maybe use these more effective therapies, you would rather try and find something that is more gentle to the patient. And so this is actually the previous slide was progression-free survival, and this is overall survival. Both are better when you combine these two therapies. So all these studies were done in DNA that was from the tumors, but then you can actually also detect mutations from cell-free DNA. And this is a really nice way of doing it, because cell-free DNA, you do a blood draw and you isolate the cell-free DNA and then do sequencing from that. So it's minimally invasive, and you can actually monitor the tumor over time. So we have actually done a small study at our local university hospital. This is this manuscript is now in revision. So we collected 70 plus patients from Kuopio University Hospital, and then took 3 samples from them at the time of diagnosis, 3 months after the onset of treatment, and then upon progression, if the patient was still around. And then we did analysis of NRF2 pathway mutations, other oncogenic mutations, and circulating tumor mutational burden. And then we had access to not all, but very many samples from these patients. These were actually had a metastasized disease, so they were not actually surgically treated. So all these samples were from biopsies. So we were able to identify quite a number of mutations in both NRF2 and KEAP1 genes, and also one pulin-3 mutation. Many of these were known activating mutations, but we also had some unknown mutations, and also some that we could sort of classified as likely loss of function, because they were residing in an area that was likely to cause defective protein. We then correlated these mutations with staining of AKR1B10, which is one of the NRF2 target genes, and quite nicely we could see that if you had a mutation, you actually also had a high expression of AKR1B10, and in some cases, for instance, this mutation was newly identified, functionally unknown, but we had very high expression of AKR1B10, and therefore it was likely that it was actually activating mutation. And so both AKR1B10 as well as NR2 activating mutations were highly sort of correlated with the survival of the patients, even with this small population. We also did the multivariate Cox regression analysis of overall survival, and quite interestingly, NR2 activating mutations were actually the highest predictor of poor survival, even more so than smoking, stage, etc. So basically this tells you that if you have an NRF2 activating mutation and you have a cancer, at the moment the prognosis is not very good. We also did a panel of other mutations, and not to go into detail, just to mention that SMARCA4 mutations were highly co-occurring mutations with NR2 pathway mutations. And also we looked into another biomarker, protein biomarker of NR2 activation, AKR1C1 expression, and we did multiplex immunohistochemistry with this, and also a panel of inflammatory markers. But just suffice to say that in addition to B1, also C1 is a very good protein marker for NRF2 activation in non-small cell lung cancer, and it's very correlated with AKR1B10. Then a few words about transcriptional signatures to identify NRF2 overactive cancers. We heard from Christina about the 6-gene panel, but we have also, others and us have also looked into some other genes. And this paper was quite recently published in Journal of Thoracic Oncology. They came up with a 46 gene score, which is quite a lot. And another, this is a Italian group that had a slightly smaller number, roughly 10, a little over 10 target genes that were phenocopied the KEAP1 mutations and correlated with survival outcomes in lung cancer treated with immune therapies. We have done also our own score. This was published in Redox Biology a few years back. What we did was that we took our own in-house gene expression data where we had NR2 overexpressing non-small cell lung cancer cells that and also control and compared that to with cells that were NRF2 was silenced with CRISPR-Cas, took those differently expressed genes, combined functional genomics data, for instance, NRF2 chip, seq data, filtered out tissue and cellular mRNA expression so that we had universal expression of the genes, and came up with a signature also of 6 genes, but these are a little different from the previous 6 gene signature, but has some common components. And this is actually pretty good in at least in cancers, small cell lung cancer, other TCGA cancers with NR2 pathway mutations, because when we compare area under the curve with known activating mutations in any of the pathway genes, the AUC is 0.94, showing that this score performs very well in these samples and distinguishes very well the cancer from the control, or the overexpression cancer from the control. It also is well aligned with the protein expression of the same genes. We see here, this is from a recent proteogenomic study where they show that many of these genes, also the protein level is highly upregulated in the mutant cancers. We took a look at the activity of again across all TCGA cohorts, and we can see that especially in lung squamous cell carcinomas and other squamous cell carcinomas, esophageal cancer, for instance, head and neck cancer, you have high activity score. And also adenocarcinoma is high up here. So this is from lowest to the highest mean score. And you can see that there's also an enrichment of mutations in those cancers. So that was about the score. So you can use also gene expression to infer NRF2 activation in cancer, but at the moment, I think it requires a tissue. You cannot really do it from blood draw. And this is one additional way of looking into NRF2 activation in non-small cell lung cancer. You can use PET tracer. This specific tracer uses a XCT, which is the same as SLC7A11 cysteine transporter, and because it's highly upregulated in NRF2 active cancers, it actually is also taking up the tracer, and so you can use this radio tracer to detect NRF2 activation in vivo. So to summarize, you can use mutational profiling of tumor tissue to detect activating mutations in KEAP1, NRF2, and Colin3. The drawback here is that especially with KEAP1, you may have mutations that have not been characterized before, and the functionality is not necessarily very clear. You have also, there's a possibility of doing sequencing also from cell-free DNA. It works very well, and because the techniques are so sensitive these days, if you want to combine this detection of NRF2 active cancers with immunohistochemistry, you can use AKR1B10 or C1 as a biomarker. They perform both very well. There are good antibodies for that. But then again, if you have tissue and you have a possibility to do RNA analysis, you can use transcriptional signatures to infer NRF2 activity. And the last example was about PET imaging that can be used for in vivo imaging on NRF2 active cancers. So I thank you for your attention, and I thank all the people in my group, especially Jouni Härkönen, who has done many of the clinical work that was presented today. Thank you.
Thank you, Annalisa, for your interesting talk. And before we move to the last speaker, we are pleased to welcome Professor Quadrato, who has just joined us. Thank you, Antonio, for being here. He will give his concluding remarks at the end of the talk. Now it's my pleasure to introduce Dr. Roberto Motterlini, Director of Research at the INSERM, the French National Institute for Health and Medical Research within the University of Paris-Saclay, France. Within the construction of NMFR, Dr. Motterlini has made outstanding contribution to understanding the NRF2 and HA1 axis and its pharmacological modulation. His critical spirit and expertise have been invaluable to the action, particularly in exploring novel therapeutic strategies targeting NRF2 and in linking basic molecular mechanisms to potential clinical application. Today, he will present his talk entitled Dietary Compounds as Activator of the NRF2 and Oxygenase 1 Axis. Please join me in welcoming Dr. Roberto Motterlini.
Thank you, Brigitta, for the kind introduction. I hope you can see my slides.
Yes, sir. All is okay.
Okay, it's a great pleasure to be here and to be the last speaker of this beautiful series of lectures that are organized through the Cost Action. And I would like to thank also once more Antonio for being able actually to organize this beautiful series of lectures and the Cost Action as well. To start my presentation, I would like to make three general statements. First of all, there is a slight change in the title. When I say dietary compounds, I mean dietary phytochemicals. And the second statement linked to that is that there is no doubt to me that looking at the literature today, there is plenty of studies confirming that the use of fruits and vegetables in the diets is very beneficial to humans. And the last statement I would like to make is that I'm not an expert on NRF2, which is the topic of these lectures, but my major focus in research is hemoxygenase. And for those of you who do not know about this enzyme, hemoxygenase is the rate-limiting step in the degradation of heme. It's an enzyme that is anchored to the membrane of the endoplasmic reticulum, looking up onto the cytosol. And hemoxygenase catalyzes the degradation of heme to liberate iron and to produce carbon monoxide and biliverdin, which in the cytosol is converted to bilirubin by biliverdin reductase. There are two major isoforms of hemoxygenase. There is a constitutive form, which is called HO2, and there is an inducible isoform, which is HO1. And it is this particular protein that we are very much interested in. H1 is a stress-responsive protein. It's an extremely redox-sensitive enzyme. And it's known now that it is a cytoprotective and it's an antioxidant enzyme. Why is that? Well, first of all, hemoxygenase degrades a pro-oxidant and pro-inflammatory molecule, which is heme. Free heme is a highly pro-oxidant compound. But equally important is that during the degradation of heme, it produces carbon monoxide, which is a signaling molecule, and biliverdin and bilirubin, which are powerful antioxidants. How is HO1 regulated? Well, I'm afraid you saw actually this scheme hundreds of times. And Christina actually made a very nice and clear introduction on how NRF2 regulates a battery of cytoprotective genes. Just to be very brief here, I just want to remind you that the NRF2 KEAP1, which is a repressor of NRF2 complex, is continuously degraded in the cytosol in normal condition. But in the presence of oxidative stress or electrophiles, this complex is disrupted, allowing the NRF2 to move into the nucleus, binding to an antioxidant responsive element, which describe not only hemoxygenase 1, but also a battery of other defensive genes, as already explained by Christina. Hemoxygenase 1, NQ1, GCLC, and GCLM are apparently the signature of NRF2 activation. And of course, hemoxygenase 1 in particular is not regulated only by NRF2, but by other transcription factors. And although I'm not actually presenting any data on BAC1, I would like to mention this transcription factor, because in reality, it's not a transcription factor, it's a repressor of the expression of a hemoxygenase 1. And the way actually this BAC1 is de-repressed is in the presence of heme. So heme is not only the substrate for hemoxygenase, but is also an H1 inducer, because when heme increases in the cells, it can move to into the nucleus, binds very tightly to BAC1, and BAC1 is removed from the RA responsive element, allowing the NRF2 to transcribe the hemoxygenase 1 gene. So how do we measure nowadays all this system? How do we measure the regulation of NRF2-dependent genes? Well, thanks to the development of genomics, transcriptomics, and proteomics approach, we've learned a lot about how NRF2 regulates the transcription and the expression of these defensive proteins. However, I would like actually to make another statement here. Assessing these omics provides no information about the functionality of a protein. So how do we measure the function of a protein? Well, in the case of a hemoxygenase, we can measure in hemoxygenase 1, we can measure hemoxygenase activity. And this can be done spectrophotometrically by measuring the bilirubin, which is the end product of heme degradation by hemoxygenase, with an assay that we developed many years ago. Sadly enough, if we look at the papers today, hardly anyone measure activity of these important enzymes. So in the next, and I have to say that back in the old days, before actually measuring the mRNA or protein expression of HO1, we used to measure the hemoxygenase activity. So in the next 20 minutes or so, I would like actually to show you how, by measuring hemoxygenase activity, I stumbled by accident on NRF2. And this is my personal story of the research I'm doing. So we have to go back to the previous century, and to be precise, to the year 1999. I was in England, and I was studying actually what was the effect of hypoxia on hemoxygenase activity. And to do that, we were culturing actually bovine aortic endothelial cells and exposing them to 18 hours of hypoxia, and then measuring hemoxygenase activity expressed here as picomole of bilirubin per milligram of protein per hour. And you can see here, endothelial cells exposed to hypoxia display an incredibly high hemoxygenase activity compared to control cells, about 24-fold increase. Then I wanted to study more in detail what is the mechanism of this activation. So I went into the literature, and at that time we knew that NF-kappa B binds to the distal enhancer region of the HO1 gene. And we also knew that hypoxia stimulates NF-kappa B activation. So the logical thing to do at this stage was to find inhibitors on NF-kappa B and see whether NF-kappa B is directly involved in the activation of hemoxygenase activity. And by looking at the literature, we found that activation of NF-kappa B is suppressed by a compound called curcumin. So I took curcumin, I exposed endothelial cells to hypoxia in the presence of curcumin, but instead of finding an inhibition of HO activity, we found a potentiation, as you can see here. There was a 40-fold increase in hemoxygenase activity in endothelial cells exposed to hypoxia in the presence of curcumin as low as 5 micromolar. Then we went to measure the HO1 mRNA expression during hypoxia. And we found that in the early time of hypoxia, there was indeed HO1 mRNA expression, which was potentiated by the presence of 5 micromolar curcumin. And then I went back to the PubMed and I typed the words curcumin and hemoxygenase, and I found 0 papers. So I said to myself, this is an interesting topic and it's quite new. So I went back to the laboratory and started to look at the effect of the effect of curcumin on endothelial cells exposed to atmospheric oxygen. And you can see here that curcumin in a dose-dependent fashion increases hemoxygenase activity at a concentration from 5 to 15 micromolar. And at higher concentration, there is a decrease in hemoxygenase activity. We found also that curcumin in a dose-dependent fashion increases HO1 protein expression and HO1 mRNA expression. And then we look at the cell viability of curcumin, and we found out that the higher concentration, you can see here, 25, 30 micromolar, the cells start to suffer. There is a decrease in cell viability in association with a decrease in hemoxygenase activity. So this is important because it's important that you measure the activity of the protein to see whether it is functional despite the fact that you have perhaps H1 protein and H1 mRNA expression. Then we wanted to determine whether curcumin-mediated heme oxygenase activation protects endothelial cells against oxidative damage. And to do that, we expose the cells to glucose oxidase. Glucose oxidase utilizes glucose present in the medium and in the presence of oxygen produces hydrogen peroxide. And the continuous production of hydrogen peroxide causes oxidative stress. And it can be seen here. When we look at cell viability, there is a dramatic decrease in cell viability by glucose oxidase. In cells pretreated with curcumin for 18 hours, the decrease in cell viability was much, much lower. And in the presence of the inhibitor of heme oxygenase, protoporphyrin 9, the cell viability was decreased even further, demonstrating that heme oxygenase is contributing to the protective effect induced by glucose oxidase. And then we expose the cells to hypoxia, as you can see here. Also, hypoxia reduces the cell viability in the presence of glucose oxidase. And this is because, but you can see that the glucose oxidase causes much less toxicity in hypoxia compared to normoxia. And this is because in hypoxia, as I've shown you before, heme oxygenase activity is already activated. In the presence of protoporphyrin 9, which inhibits heme oxygenase, during hypoxia, we have a decrease in cell viability. But when cells were exposed to hypoxia in the presence of curcumin, we have a much less reduction in cell viability. And this is again because in the presence of hypoxia in curcumin, you have a potentiation of heme oxygenase. So we published this paper, curcumin, an antioxidant and anti-inflammatory agent, induces heme oxygenase 1 and protects endothelial cells against oxidative stress in the year 2000. At the same time, what is curcumin, first of all? Curcumin is a natural substance. It is extracted from the rhizome of Curcuma longa Linn. It is a major yellow pigment in turmeric, curry, and mustard. But most importantly, when you look at the chemical structure of curcumin, it contains two electrophilic alpha-beta unsaturated carbonyl groups, which is shown here. And this is the electrophilic moiety, which is important actually in the activation of NRF2 via the binding to the KEAP1 molecule. And to remind you, an electrophile is a chemical species that accept an electron pair when forming bonds with nucleophile, such as sulfur or thiol groups, as present in KEAP1, the NRF2 repressor. At the time we were doing the experiments with curcumin, a paper by Javed Alam at the Louisiana University came out entitled NRF2, a Kaplan-Collar transcription factor, regulates induction of heme oxygenase 1 gene. So when this paper came out, I contacted Javed Alam and I asked him to explain to me how the heme oxygenase 1 gene is regulated. And what he did, he sent me a cartoon that is represented here. It's a very rudimentary map of how the H1 gene was regulated, at least at the time, what we knew, how the H1 gene was regulated. So P here is the promoter region, P is the proximal enhancer, and here is the distal enhancer region of the H1 gene. And you can see here that these regions are bounded to different transcription factors. You have NF-kappa B here, you have the heat shock factor here, you have the hypoxia responsive element, and of course you have the stress responsive element of antioxidant responsive element, which is the region which is recognized by the NRF2. So in the meantime, I was continuing my work on curcumin, and we were actually looking at the effect of curcumin in another type of cells. These are PK1 renal epithelial cells, and we exposed them to increasing concentration of curcumin. As you can see here, there is an induction of H1 in a dose-dependent fashion. And then at the time, we bought some antibodies from Santa Cruz Biotechnology, which were not working extremely well, but they were enough actually to show that indeed NRF2 protein expression was induced by treating the cells with curcumin. So at this stage, I've asked Javed Alam in the States whether he could do some experiment to prove that the expression of H1 by curcumin is effectively directly related to NRF2. So what he did, he transfected our epithelial cells with different H1 promoter luciferase constructs. And you can see here that if he transfected actually the cells with the full sequence of the antioxidant responsive element, or alternatively to the distal region E1 or E2, you could have luciferase activity showing that H1 was induced. But the removal actually of the full length of the sequence recognized by NRF2 was not induced by curcumin. So with this paper, we show that curcumin activates the heme oxygenase 1 gene via regulation of NRF2 and the antioxidant responsive element. So by searching for NF-κB inhibitors, we found by serendipity that curcumin is a new NRF2 H1 activator. So I went back to the literature and asked myself whether other NF-κB inhibitors known at that time also are activators of the NRF2 H1 axis. And in PubMed, I found another compound, caffeic acid phenethylester, which chemical structure was very similar, as shown here, to the one of curcumin. So you can see here the electrophilic element that is present in curcumin. And what is CAPE? CAPE is an active component of propolis. It's derived from the bark of conifer trees and beehives. And as I said, it's the active component of propolis, a resinous mixture that honeybees produce by mixing saliva and beeswax. And it is known for its antioxidant, anti-inflammatory, and anti-carcinogenic activities. So we tested actually CAPE, and you can see here that also CAPE was able to increase dramatically heme oxygenase activity in astrocytes in this case. And this was associated with an increase in H1 protein expression. And also, when we look at the NRF2 expression, we could see actually that in a dose-dependent manner, CAPE induced NRF2 expression in these cells. And as a third actually example of activators of H1 taken from the diet, I would like actually to tell you a little bit about chalcones. Chalcones are secondary plant metabolites belonging to the flavonoid family. They have a 3-carbon alpha-beta unsaturated carbonyl group attached to 2 aromatic rings, namely A and B. And chalcones are abundant in several edible foods, primarily fruits and vegetables, such as citrus, apple, tomato, beans, shallots, and licorice. And what we did with the chalcones, we did a study on a structure-activity relationship. So we took different chalcones that differ from their chemical structure, you can see here, and we tested them on macrophages in this case, and we look at heme oxygenase activity. And you can see here that CH25, chalcone 25, was increasing HO activity in a stronger way compared to the other two. And we realized that the methoxy groups that are present in chalcones determine actually the potency of these compounds as heme oxygenase activator. So by increasing the number of methoxy groups on the rings, you can have a more electrophilic activity on this part of the molecule, inducing more HO activity. And then to prove actually that the electrophilic moiety is really crucial in determining H1 induction and the increase in heme oxygen activity, we modified chemically the alpha-beta unsaturated carbonyl group. And you can see that in all these derivatives that do not have the electrophile, lack the ability of increasing heme oxygenase activity. So removing the electrophilic group from chalcones completely prevents their ability to activate heme oxygenase. Now let's go back to curcumin and back to the 1990s. Already at that time, there were plenty of studies showing that the curcumin has pleiotropic therapeutic benefits. And there was a list of diseases that could be, in preclinical model, be treated by giving actually curcumin. But there was actually a big problem with the use of curcumin. And this problem is related to the poor oral bioavailability of this compound. And also the poor solubility, low permeability, low absorption, and the rapid metabolism with a short elimination half-life of 2 hours. So I started to look at the literature whether there is a way actually to improve the bioavailability of curcumin when given orally. And incidentally, I came across to this very interesting paper entitled Influence of Piperine on the Pharmacokinetics of Curcumin in Animals and Human Volunteers. And in this study, they showed that concomitant administration of 2 grams of curcumin and 20 milligrams of piperine to humans markedly improves the bioavailability of curcumin, leading to a 2,000-fold increase in serum concentration with no adverse effects. So I started to look at whether we could actually use curcumin in combination with piperine and see whether we can have a higher activation of HO1 and do some study also to see whether there is a protection against some pathological conditions. And luckily enough, at that time, there were actually some of dietary supplements that were commercially available, such as curcumin 95, which contains curcumin 75 milligrams of curcumin. And then there was another actually dietary compound called super curcumin, which incidentally had a curcumin in combination with piperine. So I went to the lab and we tested actually these 2 compounds, the curcumin and the curcumin in combination with piperine, and look at the heme oxygenase activity. And you can see here that the super curcumin with piperine, induces HO activity much higher than curcumin alone. So we wanted to test whether by giving actually curcumin orally, super curcumin orally to rats, we could see whether it protects against myocardial reperfusion injury. So we dose actually Sprague Dawley rats with 100 milligrams per kilogram orally daily for 7 days. And then we extracted the heart and we placed it into an isolated system, perfusion system. We submitted actually the heart to 30 minutes ischemia and 60 minutes of reperfusion. And here are the data. So here we measure the left ventricular function pressure of the heart. And the control is here in red. You can see that when you do ischemia and reperfusion, the heart recovers only by 50%. But in the animals actually treated with super curcumin, the contractility of the heart is significantly increased to 75%. And when we use actually the heart treated with super curcumin in the presence of the heme oxygenase inhibitor, we completely actually ablated the protective effect induced by super curcumin, indicating that heme oxygenase 1 is the contributing to the protection induced by super curcumin. So I did not actually look into the literature and follow how actually the potential development of curcumin as a drug is progressing. But a couple of years ago, actually, I did so. And to my surprise, actually, there is a lot of studies on trying actually to improve with different formulation of curcumin, the bioavailability, the absorption, and the cellular uptake of this natural compound. Although the number of clinical trials in this case is extremely low. But in essence, this formulation of curcumins improved dramatically in the last decade. And there are a lot of diseases that have been tested with this new formulation of curcumin, including cancer, chronic kidney disease, Crohn's disease, non-alcoholic fatty liver disease, in many of the diseases, we can see that the oral bioavailability of curcumin increased from 11 nanogram per milliliter back in the 90s to 627 microgram per milliliter to today. These curcumin formulations were found to be safe and well tolerated at higher doses ranging from 2 grams to 12 grams per day, and for a prolonged duration of 6 months to a year. And finally, just to mention that a few years ago, in 2012 to be precise, we did a comparative assessment of known NRF2H1 activators that were known from the literature at that time. And we searched and tested actually about 100 compounds that were capable of activating NRF2 and H1. And we systematically actually measure these compounds in microglia for HO1 activation, hemoxygenase activity, their toxicity, and their potentially anti-inflammatory effect. And the best compounds, to summarize this study, that came out were carnosol, dimethyl fumarate, and, believe it or not, supercurcumin. It's a pity that the data on supercurcumin are unpublished today. We haven't been able actually to continue this study for various reasons. But still, I think curcumin has a potential actually to be used actually as a dietary compound to activate NRF2 and HO1 in tissues and in pathological conditions. So to conclude, some reflections and perhaps a take-home message. What I've showed you today is that, of course, when we test natural compounds or any activators of NRF2 and HO1, we usually actually test one natural compound at a time. There is no indication in the literature what happens when you use a combination of dietary compounds, and in this specific case, phytochemicals. Does the combination of many phytochemicals is able to activate NRF2? Why is this important? I think it's important because I go back actually to the first statement I made at the beginning of my presentation, that a diet rich in a diverse array of fruits and vegetables has been consistently associated with reduced risk of chronic diseases and increased health span, ultimately adding healthy years to your life. Now, if we look at actually at the estimated number of phytochemicals present in different fruits and vegetables is about 10,000. Now, I do not know precisely how many of these phytochemicals could be NRF2 inducers. We don't know that. But I'm pretty sure that even if these compounds, these phytochemicals, the bioavailability of these compounds is very low, we need actually to test the combination of phytochemicals. We do not know whether there is a synergistic effect by adding different potential inducer of NRF2 together. So this is a study that should be done, in my opinion. Until then, the most humble suggestion I can give you is that you keep eating fruit and vegetables because they are good for you. And maybe if you are lucky, if some of them are, some of the compounds that are present in these fruits and vegetables are bioavailable, you could have NRF2 activation and protect yourself from oxidative stress and perhaps different diseases. And if you decide actually to introduce curcumin in your diet, please do not forget to add some piperine into it. So add some black pepper. And with that, I would like to thank actually my current group, and particularly Dr. Roberta Foresti, who is my long-lasting collaborator. And I would like to thank also the pre-omics groups, so the people who actually did the work and the data I presented to you today on the effect of phytochemicals on hemoxygenase, Elizabeth, Philip, Reka, Martha, and Giovanni. and also Javed Alam, whom has been able actually to demonstrate that indeed curcumin mediated induction of H1 is via NRF2. And thank you for listening.
Okay, thank you, Roberto, for your interesting presentation, for sharing your experience with us, very inspiring. Thanks to all our speakers for keeping perfectly on time. We received a lot of questions in the chat, so we can start to read the first question for Christina. Okay, Christina, are you here? Yes. These are gene expression, and I was wondering if we could consider molecule as biomarker, glutathione or others, of NRF2 action. Thank you, Roberta.
Thank you, Roberta, for this very valid question. I mean, considering that NRF2 can induce the expression of two key enzymes that are important in the synthesis of glutathione, why not use glutathione as a readout? Yeah, and I think people are using these assays to determine either total glutathione or also the ratio of oxidized and reduced glutathione levels. But the question is really, does glutathione show direct target engagement of NRF2 activity? And my impression is, or my feeling is, that it's kind of an indirect measurement, not directly showing NRF2 signaling engagement. But of course, there might be other molecules on top of these transcriptional or protein signatures that might be relevant.
Well, maybe how, like you did with the genes, maybe it's a matter of a combination of biomarkers that together could be more predictive.
Exactly. I think maybe this, I'm of the opinion that maybe this 6 gene protein biomarker is too simplistic. So there are, for example, maybe metabolites that are important and could be taken also into consideration for measurements. And yes, it's complex and definitely also dependent on the context, on the disease context, on the timing of the disease, or if you're measuring drug activity. So there is a lot of layers to that.
Okay.
Thank you.
A similar question is from Alessandra Berri from Istituto Superiore di Sanità. She say your panel is very compelling, but why this 6 specific gene? For instance, could a simple panel, just the top of 2, be sufficient, more cost effective for clinical use? And a second question is, given the influence of factors like diet and lifestyle, how do you propose to establish a baseline range for this panel?
Okay, so we have focused on 6 because our rationale was that maybe to have more biomarkers, is a more robust signature than relying only on 1 or 2 biomarkers. So this panel of 6 could be more robust across different disease settings as well. And on the other hand, HMOX1 and NQ1, they probably only emerge because due to some bias in the literature, because this is what people just have already traditionally used, and it's been used now then also in my research, and this is how it is propagated. So there might be that bias as well. Yeah, so I think more biomarkers for this complex regulated.
You have a lot of questions, then I can send you by mail, try to reply personally. But the last question for you is from Vladimir from Ukraine. He say, thank you, Christina, for the nice talk about NRF2. Have techniques for its monitoring well standardized, and which approaches were used in this case? Thank you, Vladimir.
So in our studies that we reviewed, most of the people used quantitative reverse transcriptase PCR, so assessing transcript levels of targets, but of course Western blots to assess protein levels have also been used. it determines whether you're assaying the biomarker at the transcript or at the protein levels. And there are difficulties with each of these two measurements. So maybe protein measurements can be more quantitative, but then they are inherent to biases in the detection method. Yeah.
Thank you, Christina.
Okay, we can move to the question for Professor Levonen. I see that you have also replied to Roberta, but now I want to make this question from Elisabetta Profumo. What are the pros and cons of expression-based versus mutation-based biomarker in different applications? And what are the potential uses of this biomarker in the clinical setting and drug discovery? Thank you in advance, Elisabetta Profumo.
Thank you for the question. I can also elaborate some more on Roberta's question, but maybe I'll answer the latter question first. So I think the good thing about the expression-based biomarker is that it's ignorant of the cause of NRF2 overactivation. So we know that, for instance, in lung cancer, about approximately 25-30% have somatic mutations in NRF2 pathway genes. But there are also a number of other mechanisms, and it has been estimated that as much as 50% of all non-small cell lung cancer patients would have elevated NRF2 activation. And the reason being transcriptional upregulation of the genes or some other epigenetic modifying co-mutations, etc. So the expression-based biomarker is agnostic of the reason why you have elevated NRF2 activity. Another thing is that you don't know whether the somatic mutation, if it's a new mutation in KEAP1, you don't really know a priori whether it's activating. If it's not in a region that is classed, I mean, if it's not in a region that has been extensively studied, and since we don't have the whole crystal structure of KEAP1. The pros of the mutations is that you can also use blood-based techniques like ctDNA. So with respect to potential uses of biomarkers in the clinical setting and drug discovery, I think it's already, KEAP1 mutations are already used as a prognostic biomarker. But the thing is that it doesn't really, even if you have a KEAP mutation and that is actually predictive of poor outcome when you use checkpoint inhibitors. Regardless of this, the checkpoint inhibitors are used for those patients as well. But now that we know that maybe these patients would be better to have a combination therapy with CTLA-4 and PD-1, PD-L1 blockers together. This is actually a really good thing that you can use the mutation as a biomarker for the success of this specific treatment. With drug discovery, of course, you have to stratify the patients, and if you don't have somatic mutations, it might be better to have the transcriptional signature. So drug discovery, I think it's really selecting the right patient for the treatment. Thank you. And just a little bit further on Roberta's questions, whether these mutations or more mutations are detected. Indeed, in most cases, there's a panel of genes that is detected from cancers, and it depends on one cancer center to another. But in some American cancer centers, they do exome sequencing, even whole genome sequencing from all patients. But I think it's a resource thing. It's quite expensive. when the sequencing costs go down, I presume that more and more patients will be sequenced for more than just a few genes. Thank you.
Okay, thank you, Cristina. I found a provocative question by Antonio Quadrado in the chat. He say, can artificial intelligence help define the good marker combination? He described, for example, for hepatic NRF2 activation. If I don't understand who the question is directed, but
I'll take it.
It's connected with Roberto on the bilirubin studies.
okay.
Okay.
Okay. So the question, if we can use artificial intelligence, correct?
So the question is, so the problem is that when you have a biomarker that you are studying in blood, for instance, a non-invasive technique, you have the problem of looking at a specific organ in a systemic situation. And I was wondering whether NRF2, because it is an ubiquitously expressed protein, can have this kind of confounding results. And the question is about bilirubin, because most of bilirubin is coming from liver, right? For instance, so the question is whether it would be possible to analyze HO1 indirectly with a surrogate biomarker like bilirubin, and maybe some other markers like transaminases and something else. And as I know that you are going to tell me that it is very specific, I introduced this issue of the artificial intelligence.
Yes.
Okay.
Thank you for the question. First of all, actually, it's interesting that you mentioned bilirubin, because when we worked on the review that Cristina mentioned and we published together in Redox Biology, on the first of the draft, I was proposing actually bilirubin as a potential metabolite. But then actually during the discussion with the other co-authors, we actually realized perhaps bilirubin, it's not really specific for HO1, because bilirubin is also, metabolized differently. It's so the way actually bilirubin is metabolized by the liver. So we cannot actually say that bilirubin production in vivo is coming essentially from H1 protein expression, it depends on the elimination of bilirubin from the organism. You see how the bilirubin is eliminated. So it's a difficult question to address. Yes, Roberta?
But maybe we could measure CO, carbon monoxide.
Yes.
But I think
it would be free bilirubin.
Sorry?
You want to measure free unconjugated bilirubin, because conjugated bilirubin, like Roberto said, is a matter of, it is metabolized. So
yes.
It depends on other enzymes. But anyway, I just wanted to have this provocative question.
Yes, of course. But now there are the studies coming out showing that we can actually see specific organs aging, in the whole organism faster with, different kind of markers. So I think the approach of AI in combination with, for example, organ aging and other parameters will help us to work in this direction, Antonio.
But nevertheless, we are very happy that H1 comes always at the top when Cristina did her analysis, and it's still very promising to me. And as Roberta said, I think it's important we progress in measuring now CO in tissues, but whether that could be possible actually maybe in circulating white blood cells in humans, we could eventually measure actually the activity of HO1, and in combination with that, of course, the production of bilirubin specifically in these cells, and also eventually the accumulation of CO. If CO is retained in the circulating white blood cells. This could be perhaps the most logical approach to have data from humans in relation to the possibility of NRF2 induction and HO1 activation.
Okay, another question from Marzia. She say, dear Roberto, great talk. Since hemoxygenase 1 is one of the most sensitive genes induced by oxidative stress regulated by NRF2, do you think it can serve as a reliable biomarker on NRF2 activation in clinical settings or during dietary intervention? This is from Marsha.
Thank you. Interesting question again. Well, I could answer by saying it seems that HO1 is really part of the signature of NRF2. And the reason, again, I go back to the biochemistry, sorry, I may be totally biased and I accept the criticism, but heme oxygen is truly an antioxidant enzyme for the reason I mentioned to you at the beginning. It removes a pro-oxidant molecule. Free heme is a pro-oxidant molecule. It cannot be free. It has to circulate in the body, bound to any protein. It's bound to hemopexin, it's bound to albumin. It's never free, because if it is free, it's a problem. And that's and that's why BAC1 is important in combination with the NRF2. It senses the changes, the subtle changes of heme. So heme is removed, and it produces actually biliverdin and bilirubin by hemoxygenase, which are the most powerful antioxidants. And plus we have carbon monoxide. So we have a synergistic effect there. So to answer to Marcia's question, I'm biased. I'm not saying that HO1 should be the only one, but it's an important one to be taken into consideration, definitely.
Thank you for your reply. Another question for you. Vladimir say, bravo Roberto, you stressed that the ancient approach to measure activity of enzyme is very important, not only model mRNA level as indicator of gene expression. Thank you. It's from Vladimir. Quindi to test, he say...
Thank you, Volodymyr. Actually, we met for the first time in Istanbul at the beginning of October. Yeah. And he was the one actually who stimulated my talk here, because actually he was the one who presented only data on activity in his talk. And so we discussed about that. And perhaps for the young researchers, it is important that we go back to the basic biochemistry and measure the activity as well. Because in combination with gene expression, mRNA expression, and protein expression, we can have the full picture of how actually these defensive proteins are regulated by NRF2 or other transcription factors. Thank you, Volodymyr.
May I add a little bit? Also, I'd like to call attention to classic biochemistry, because always we deal with steady state level of intermediates, of proteins, etc., etc. Actually, you mentioned this, that it is bilirubin appears from enzyme, it disappears. So we can measure steady state level. It's also basic biochemistry. Thank you, Roberto.
Thank you.
Okay, another question for you, and this is a very critical question from Tuomas Tikkanen, University of Eastern Finland. Thank you, Roberto, for the very interesting presentation. Curcumin is a no-painless interference compound. Can you be certain that curcumin really acts as a hemoxygenase activator, or can this be an artifact due to the curcumin intrinsic tendency to give a false positive in a large array of different assays, in addition to its promiscuity? It's a tendency to bind protein non-selectively. So hemoxygenase activity is an artifact induced by curcumin, is an artifact, or is a specific effect?
Thank you for the question. I'm sure at 2000% that is not an artifact, because we measure the activity. It can't be an artifact. Actually, when you treat the cells with curcumin and you extract actually the microsome, and by the way, the hemoxygenase activity assay, it's a very tedious assay. It can be done, but it precisely measures the activity of this protein. to the average standard of, forgive me if I say that, of postdoc and PhD students, it's a challenge to get back to the laboratory and set up actually the heme oxygenase activity. But if you do that, and I wish actually you could do the experiments yourself, I can guarantee you the curcumin, calcones, any kind of phytochemicals that we tested in the lab are inducer of HO1 and increase heme oxygenase activity. It is not an artifact.
The last question, then we have to make the conclusive remarks. So the last question for you, Roberto, is from Hamad. He says, thank you, Roberto, for this great presentation. Do you think enhancing curcumin bioavailability necessarily translate to stronger NRF2-mediated cytoprotective effect, or could there be a saturation point in the pathway? You say you mean not just following the rule, more bioavailability equal better effect to know how much we can increase our uptake. So he talk about curcumin bioavailability, he tends to a plateau, or you can increase much more the curcumin bioavailability and effect.
Thank you for the question, Ahmad. Actually, I have to say that Ahmad is a postdoc in my group. So he's a new one, and he asked me the question. So a good question, actually, and I would like to answer in this way again. When we measure activity, the heme oxygenase activity that I presented to you today, with the dose-dependent increase in heme oxygenase activity of curcumin, you could see that there is a dose-dependent increase up to a certain concentration, and then there is a drop in the HO activity, which start to coincide with the enhanced cytotoxicity. So it's a critical question. We have to find the dose that increase heme oxygenase activity, but then does not cause toxicity. So we have to find the balance. And in vitro, it's perhaps easy, but the difficult part is in vivo, when you have to deal actually with bioavailability. So maybe we can discuss more in the days to come, and we can eventually design some experiments to address more precisely this question. Maybe, Ahmad, you can do some experiments for me to address your question.
Thank you.
Yeah, for sure.
Okay, thank you, Roberto, for and I want to thank to all our speakers for the interesting talk, the clear answer, and thank you everyone for participating in this webinar. I don't know if Antonio want to say something. This is the today marked the final webinar. Is a successful 4-year series organized in collaboration with the Biomarkers post-action, coordinated by Antonio. So I want to sincerely thank you for your support, and if you want to say something, what I say in the I also want to extend, you are pleased to share that the new webinar will be launched the next year, and so we would invite all of you to suggest topic and a speaker to maintain light in the most relevant excited development of NRF2 research. So we are here, we are happy to collaborate again with this format and by using this platform. So you can conclude them.
Thank you very much, Brigitta. I just want to say very quickly, I want to thank the speakers. I want to thank also the people who are here. And I particularly want to thank you and Professor Luciano Sasso for running these excellent webinars. Even though the cost action is over, I really hope that you can keep going on with this excellent activity. So thank you very much.
Okay, thank you so much, everyone, for participating in this webinar. The webinar, I want to remind that the recorded webinar are always available on website antiox.it. So share this information with your colleagues and students, and I'm looking forward to keep you in touch for next events. So bye-bye.
Thank you. Bye-bye.
Bye-bye. Thank you very much.
Thank you. Bye.
Bye.
Bye.